Okay, good morning, everyone. Many thanks for the nice introduction and for having me today. So I'm now talking about our work in accelerating the discovery of new antiviral molecules. And yeah, the talk is called On the Automation of de novo Molecular Design and Chemical Synthesis Planning. It's a case study on SARS-CoV-2, but the methods we're showing here, they're not limited to SARS-CoV-2 or also not very specific to this pandemic. So let's see whether this works. Okay, excellent. So traditional approaches to developing novel materials and molecules are time-consuming and costly. So for example, it takes approximately 10 years and costs 2 to 3 billion USD to bring a new drug to the market. And from all the investigated drug candidates, only about 0.02% survive the cascade of clinical trials and safety checks. So effectively, the cost to develop a new drug are roughly doubling every decade since the 1950s. And now in the last decade, the hopes are high that machine learning can accelerate the discovery and reduce these costs. And in our research, we leverage different types of biological data, such as protein, cellular, omics data, and then we develop deep predictive and generative models to add the design of new molecules. And the overarching goal is to close the loop and automate all parts of early discovery. So this is 4 main components: the generative model, which basically proposes a hypothesis of molecules with desired properties that you can see at the top. Then automation of chemical synthesis and the automation of the experimental validation, as you can see at the bottom. And last, the data, of course, and the data that is acquired through the experiment that can then be used in order to actually close the loop and update and improve the models that you're developing. So when we began this project in spring 2020, it slowly became clear that we do not have very effective drugs to treat acute cases of COVID-19. The strategy of repurposing approved drugs such as remdesivir or hydroxychloroquine turned not out to be very successful. And we therefore decided to build on our previous work on deep generative modeling for cancer drug discovery. And we tried to sketch a pipeline for automatic and accelerated discovery. So let me make one thing very clear here. The goal was not to develop a new COVID-19 drug. That would be a moonshot project far beyond our skills. But the goal of the project was to exemplify how the discovery process of new molecules in general can be accelerated and automated so as to minimize the amount of human intervention that is needed. So let's dive into the content. Here's an overview. So drug discovery is usually a process that starts with a protein target to which the molecule should bind, the molecule that is going to be devised. So the first component of our pipeline is a deep conditional generative model that can be conditioned on a protein target, and it should produce molecules that bind with these targets. But the objective of this work was to automatize the pipeline all the way until we have to go to the lab and create the molecule. So this includes the component that you see in the middle now, which is about finding synthetic routes via retrosynthesis prediction models. So this is a model that provides us a list of precursors that we need to purchase in order to synthesize a new molecule. But it doesn't tell us how the reaction can be executed in the lab. And for that purpose, we use a last model for synthesis protocol generation, which converts the synthesis tree into a stepwise execution plan, as you can see on the right. Let's now start with the first component, the conditional molecular design. And if we formulate this problem from a reinforcement learning perspective, we could say that given a context C, so this could be a protein target, the goal is to maximize the probability to generate a molecule that produces a maximal reward. So that sounds quite abstract, but the intuition here is that for one context, so for one protein, we want to sample in one part of the chemical space to produce new good molecules. But for another context, we want to sample somewhere completely else. And we want to learn to map each context to the right part of the chemical space for the sampling. The reward function we are maximizing needs to depend on the context, but otherwise there are few requirements. For example, it can be multi-objective, and in our case, it is a combination of an affinity score between the protein and the molecule and a penalty for toxicity. So Here's the illustration of the process. We first train two separate variational autoencoders. So those are generative models, one for proteins and one for molecules. They're trained unsupervised and independently on large-scale database, namely UniProt and Campbell. And once these models are pre-trained, we preserve only the protein encoder and the molecule decoder. And then our conditional generator is what you can see in blue on the left side of the slide. It's a hybrid VAE, so to speak, that encodes from one modality and decodes to a molecule. So in other words, we encode a protein of interest. We exploit the fact that we fix the latent spaces to have the same size, and then we can decode a molecule from this protein encoding, basically. So we convert a protein to a molecule. And this molecule is then passed to the reward function that you see on the right, called critique. This reward function is performing a virtual drug screening, asking whether the generated molecule can be expected to bind with the target of interest. And the outcome of this virtual screening is then used as a feedback to update the conditional generative model. But this critique or reward function is itself a deep learning model. And this is a drug target affinity prediction model, which consumes two inputs. There is a protein target, as you can see on the left. It's represented by the amino acid sequence. And on the right side, you see the molecule, which is represented as SMILE string. So this is this text-based chemistry language. And it then applies, this model applies several 1D convolutions to basically aggregate information in local substructures. And then there is a contextual attention mechanism, which makes the model more interpretable. The output of the model that's important is a binary classification, which indicates whether or not the molecule binds to the target. And we trained this model on about 2.7 million pairs of proteins and ligands from BindingDB. We find that we can distinguish well between binding and non-binding pairs. And we also find that we roughly preserve the promiscuity of the protein targets as present in the data. So this made us sort of confident that we are not systematically under or overestimating how reactive some of the proteins of interest are. So let's go ahead now with the conditional generation. For that task, we extracted 41 targets, SARS-CoV-2 related protein targets from a Nature paper from summer 2020. This included viral as well as host proteins. And the question we raised then is, can we de novo design compounds with high binding affinities for unseen COVID-19 protein targets? And we performed a leave one out cross-validation scheme with this reinforcement learning optimization that I was showing before. And as a reward function, we used a combination, as I said, of binding affinity and toxicity, both predicted by our own models. So the results show that we generate more binding ligands or molecules in the vast majority of cases. There was an average growth of 83% in the number of molecules that are predicted to bind. And here are two examples, the affinities on the x-axis, and we can shift these baseline distributions that are shown in gray toward the right side, which means that we sample more molecules that are predicted to bind or that have high affinity with the target of interest. We did not, however, succeed in our secondary objective, which was reducing the amount of non-toxic molecules, but it's a secondary objective. And the key thing to note from this result is that this is the first evidence of a conditional molecular generator that can generalize to unseen protein targets. And this means it can produce molecules with high affinities for targets it has not been exposed to. So one problem with novel ligands is cross-reactivity. A drug candidate can be expected to bind with at least a dozen different targets. And with our affinity predictor, we assessed the cross-reactivity of 3 sets of molecules, optimized, shown in green, unbiased in red, and random drugs from Campbell in blue. And if we focus on 500 non-viral proteins, we find that the optimized molecules are significantly less cross-reactive than random drugs. And if we focus on these relevant targets, on these 41 SARS-CoV-2 targets, we find the reverse effect, here our optimized molecules are significantly more cross-reactive than unbiased and random molecules, which is exactly what we want. And this is remarkable because we didn't explicitly optimize for this selectivity. We then compiled a set of 20,000 conditionally generated molecules. We decided to synthesize one candidate, and we used a bunch of property filters to nail down the list and estimated retrosynthetic trees for the best 250 candidates. And for this retrosynthesis prediction, we used the models from our colleagues that are deployed in the RxN service, the IBM RxN service. And the goal of this model is to find a set of commercially available starting materials that can be used to synthesize the molecule. This retrosynthesis prediction was successful in 30% of the cases, which left us with 74 molecules. And then we selected one for the synthesis. This one was 3-bromobenzolamine, which you can see there at the top. It's a full substructure. It's a fragment of abidol or umifenovir. And abidol is a broad spectrum antiviral drug, which somehow decreases mortality in COVID-19 patients that are already hospitalized. It's not used in Europe, but it's used a lot in Asia, actually. And the mechanism of action of abidol is that it is a virus host fusion inhibitor. So it blocks the ACE2 receptor, which sits on the human epithelial cells, and this prevents the viral entry to the target cell. Now, the key observation here was that 3-bromobenzolamine was generated by our model specifically to target the ACE2 receptor. And we therefore hypothesized that 3-bromobenzolamine could operate in a similar mechanism of action like abidol, especially since we found evidence that the presence of bromine is key for the efficacy of abidol. So after we opted for this molecule, we choose a one-step reaction using a nitril reduction. We used another model from our colleagues, the smiles to action model, which converts the reaction smiles that you can see on the left to a sequence of experimental steps that need to be done in the lab, like adding certain components, steering, these sorts of things. And then the reaction was successfully executed in our lab in Zurich. So unfortunately, we don't have experimental validation of that molecule on the ACE2 receptor. But as I said in the beginning, the overall point of this paper, of the project, was to show a proof of concept on the automation preceding the wet lab synthesis. So what's next? As you might have heard, the IBM lab in Zurich last year launched RoboRxN, the first cloud-based AI-driven chemistry lab. This is a robotic platform based on an integration of AI, cloud, and automation technologies that enables to plan and also execute chemical reactions remotely. We actually verified the synthesis of 3-bromobenzolamine using this robotic platform. And here's a short video where you can see this platform in action. I hope that the video is playing. Okay, great, the video is playing. I'm not a chemist myself, so I don't really understand what is going on here. But what you can see there's a robot, and you can read there's a new molecule currently being created or synthesized, and therefore at the moment one ingredient is being added. And with this automatic platform, we basically hope to move one step closer to closing the loop in the accelerated discovery of new materials. And yeah, with this, we arrive at the end of the presentation. For the project I presented, all code, data, and the pre-trained models are available via the GitHub repository, Pacman SARS-CoV-2. That's also linked in the paper. And yeah, with this, I would like to close, and I would like to obviously thank all my amazing collaborators who have had a major role in making this work possible from IBM Research. And the paper is published in Machine Learning, Science and Technology. You can read all the details there. And now I'm happy to answer your questions.
Okay, thank you very much. So as you said, your method is very general and it's more like a proof of concept for SARS-CoV-2. So I was wondering how close you are to identify the antivirals for SARS-CoV-2. And so I've seen that, if I understood well, that you restricted the list of proteins that are relevant. So what would be the next step? The next step would be some experiment, or you think still here machine learning would be useful to understand even a bit more what would be the potential antivirals?
Yes, so the next step would certainly be to perform some experimental validations of the molecule that we synthesized to verify whether it indeed binds to the ACE2 receptor. This is a bit beyond the scope of this specific project because as I said, the goal was rather to exemplify how this whole pipeline can be automatized in principle. And we are not planning to basically enter this race of like COVID antivirals or so. But we have now other projects where we are focusing on more specifically on diseases where we have more domain knowledge also and to find new drugs for these diseases. So the main point here is like to exemplify the automation, and we did this by taking an example, SARS-CoV-2.
Okay, so if I understand well, your main contribution is this automation. So I was wondering, in terms of computational cost, how intensive it is?
Yeah, that's an excellent question. So I would say that there are many components in there, and these models need to be pre-trained before they can be used for inference. So these are deep learning models. They're usually trained for a couple of days, but not a week. So on GPUs that cost maybe a few thousand bucks, but not tens of thousands of euros. At inference time, then these models are relatively quick in the sense that you can get results within minutes from the model if you're, for example, sampling new molecules or if you're evaluating certain properties.